Hello, my name is Stefan Bram, CEO of Ncardia, and it's my pleasure today to talk about the company. Ncardia is a company focused on drug discovery and cell therapy using iPS cells. So our mission is to implement that iPS cell technology in drug discovery and cell therapy to get better therapies to patients faster. We are globally orientated with main facilities in Europe, in Leiden in the Netherlands, in Gosselies in Belgium. So the Ncardia team has 10 years of experience in the iPSC technology, close to 50 people. 42% of them are PhDs, and we are working on manufacturing of iPSC-derived cells, manufacturing process development, drug screening, quality, and sales and marketing. What we are doing in those drug discovery and cell therapy segments is really taking iPS cells, manufacture differentiated cells at scale, and using a variety of assays to characterize and test either with and without molecules. So we are working in drug discovery and cell therapy, which at first glance might look different, but if you think harder, there are lots of parallels. So in both segments, we are starting with humans. We are generating iPSC. We are manufacturing the cell of interest. In drug discovery, we are obviously developing assays to measure compounds effects and try to do that in a high throughput screening manner. But we see that a lot of those assays are directly useful for cell therapy as well, because there you obviously want to make sure that after you have developed a cell type, that you understand the functionality, the potency, the efficacy. And we see, in fact, that a lot of the assays developed for drug therapy are directly useful for cell therapy. So there is a lot of crosstalk and synergy between those segments. So to go a bit deeper in drug discovery, so the key benefit that we've developed at Ncardia is that we have developed proven cell models and brought together all key technologies for disease modeling and drug testing. So to start off with the classical drug discovery funnel, which is depicted here. So I don't want to bore you with details, but obviously it's all about decision making. And the good thing today is that we see that the majority of companies are using iPSC technology in some way, in some form during that process, typically to generate human proof of concept a little bit later in the process. With Ncardia, we try to bring that step up, and we try to implement the human iPS technology as early as possible to actually maximally get the benefit of it and use that human biology to identify molecules that should work in human. For this, we are focusing on cardiovascular and CNS diseases. And the types of projects we are doing are basically spanning between target discovery and validation, primary and secondary screening, hit to lead and lead optimization, and preclinical development. So in target discovery, it's very often trying to validate a target, for example, through phenotype rescue. In primary or secondary screening, it's typically phenotypic screening, trying to identify a molecule that reverses a disease phenotype. In hit to lead and lead optimization, it's typically trying to establish SAR and to measure potency. And in fact, that can also be for safety. So we've seen projects that progress to preclinical or clinical phases where a safety liability was identified, and where basically we rescreened the lead series to try to identify a clean variant. And then in preclinical development, we are helping companies with patient stratification, putting in different genetic backgrounds in the test system to identify where the molecules works best. So at Ncardia, I already mentioned, our focus is on cardio and CNS. In cardio, we have ventricular cells, we have atrial cells, we have endothelial cells, we have smooth muscle cells. In CNS, we have astrocytes, we have CNS neurons, peripheral neurons, cortical neurons, and microglia. We've brought that together with our disease modeling capabilities. So we're using CRISPR-Cas to either introduce a mutation or to repair a mutation. We are typically using in our projects chemical induction of disease phenotypes to generate a larger assay window and to be able to screen for that. And we're doing a lot of physiologically relevant co-cultures to mimic the biology. For example, co-culturing microglia with neurons, astrocytes with neurons, fibroblasts with cardiomyocytes, etc., etc. And then on top of that, we've built a whole panel of assays ranging from impedance, electrophysiology, gene expression, calcium transients, biomarker assays that are typically on plate radar technologies like HDRF or alpha LISA. We're doing high content screening, metabolism assays. So basically everything you would expect in a state-of-the-art drug discovery lab. So to bring that together and to summarize that, so the idea is we are starting with patients, developing iPSC-derived cell models for the human disease. We're using our manufacturing technologies to manufacture cells at scale, develop functional assays to identify the appropriate molecules, bring that to high throughput screening, ideally to identify a molecule that can go back in the patients. And where Ncardia really makes a difference is in building better cell models in cardio and CNS, doing everything at scale so that we can actually manufacture enough cells for screening, that we making the assays robust enough for screening, and helping companies with stratification. So introducing different type of mutations, sourcing different IPS lines, and repeating the process with a panel of lines. So from this, I'm going to shift gears a little bit to cell therapy. So here, the focus obviously is not on the molecule, but more on the cell. And the interesting thing is that what we are doing here is applying the knowledge that we have been building up the past 10 years on a variety of different cell types. So whereas we are focused on cardio and CNS in drug discovery, when we work with cell therapy clients, we work across all kind of different lineages. And we basically apply the developmental knowledge that we learned in cardio and CNS on all kind of different cell types. So no restrictions in terms of cell types. Focus really is to help companies develop a scalable manufacturing process. So, before I go into details, I just want to spend 1 minute on technology that we are typically using for this. So when we're looking at literature, we see a lot of small dishes. And in these small dishes, we typically see that you see these technology S-curves. So at some point, it becomes very, very hard to scale such a system out. And you reach a technology limit. And so with 2D monolayer dishes, you reach that pretty fast. When we realize that, we shifted to multi-layers, and at some point you will reach that as well. So more recently, we have developed our processes on controlled stirred rate bioreactors, and in our hands, that's a technology where we can really start to generate large lot sizes. So for cardiac, for example, we are now routinely doing 10 to 20 billion cells a run, which is quite a bit more than what we could have done a couple of years ago. To give you a little bit of details, the way it looks like, so we're starting with iPS cells. Those iPS cells are steered in the mesoderm direction. The mesoderm is patterned to become cardiomesoderm, and from the cardiac mesoderm, we get cardiomyocytes. So we cryopreserve those cells. When we stain them, we see the cardiac markers. When we do flow cytometry, we see the cardiac markers. When we do electrophysiology, we see the normal functionality of the cardiomyocytes. So to look a little bit deeper in our in-house data, so this is the manufacturing cost on the y-axis in 2017 of generating 1 million cells. And basically what you see, we're spending a lot of money on materials and on labor. And in 2018, we optimized using the same technologies, and we basically got close to 2x better. And that's basically an effect of more experience. What we did in 2019 was shifting gears and going to controlled rate bioreactors, so controlled bioreactors. And what we see now is that we massively reduced materials, labor, and facilities. And in fact, it's a 16-fold reduction of cost with a 50-fold increase in yield per FTE. And so basically what happened is that we now do with 2 people in less than a month what we did with 6 people in a year in 2017 to 18. And you basically see that here as well. So the maximum annual yield of sales per FTE is now grown to 100 billion, showing that we really get into the numbers that you would need for cell therapy. So the way we work with cell therapy partners is really from concept development up to preclinical development. So we start projects on literature searches, on FTO and IP analysis, on scientific evaluations, on making the development plan, up to developing the cell culture system, developing the quality control methods, developing the assays, sourcing materials, establishing small-scale manufacture, identifying critical process parameters, scaling that up, assessing robustness, doing pilot runs, optimizing cryopreservation, building the production documents, so the batch control records, the batch records, the SOPs, and up to the point that the process is ready to be tech transferred to a GMP facility. So we are really uniquely positioned in the market in that early phase, developing processes, bringing it to a GMP facility. So to summarize that, Ncardia is a stem cell solution provider working with partners in drug discovery and cell therapy. We have in-depth knowledge and experience to develop and validate predictive assays, and we use that in drug discovery for both safety and tox projects. With everything we do, we are focused on scalable solutions with relevant assays up to high throughput screening, and we are uniquely positioned in the cell therapy space by leveraging our 10 years of experience providing commercial products at scale. With this, I would like to thank you for your attention. If you have any questions regarding projects in drug discovery or cell therapy, more than happy to talk to you. Don't hesitate to reach out to us at support@Ncardia or through our website, ncardia.com. Thank you.